This method can help answer key questions such as how to positively modulate the activity of a gene by taking advantage of mRNAs that are present in the cells and tissues, how to compensate for deficiency of gene activity, and how to achieve high translation from mRNAs in vitro and in vivo.
The main advantage of this technique is that we can broadly screen SINEUPs target genes in living cells by imaging without fixation and collection.
So the implications of this technique extend to our therapy of haploinsufficient diseases because SINEUPs can induce twofold increase in the deficient protein, such as frataxin in case of Friedreich's ataxia.
To begin, open the Zenbu browser and click on Phantom Project Database of interest. Search the target gene and check for transcription start site, or TSS, and expression level in desired cells and tissues. To determine TSS, consult the example analysis of Parkinson disease protein 7, or Park7 mRNA. Check TSS by cage peak region. To determine cell and tissue-specific transcription expression levels, select the cage peak region and click on Magnify Genomic Region to selection. Check Park7 expression in the cell and tissue type of interest by exploring the transcript expression list at the bottom of the page. Then design binding domain sequences of several different lengths, corresponding to 40 bases upstream and 32 bases downstream of the first methionine or AUG. Before transfection with SINEUPs, seed cells of interest on 2 poly-D lysine-coated 24-well plates as described in the manuscript.